real fast, for those of you who are online, tell us your name, where you're from, and briefly why you're interested in this area. Start with Anne.
I think that some of them don't have
They don't all have microphones?
Yeah.
All right. It looks like,
Michael.
where's Michael?
Yeah.
Michael does, so let's start with Michael. Michael Seifert, you want to tell us where you're from? You may not be right there.
Okay.
Okay. We'll count him as an attendee anyway. Who is HTS? I don't know if they've muted themselves. We should see if it's muted.
Well, maybe we'll ask them to type into the...
Yeah, we'll have a record of it, I think, because that's a nice big list online, and we do need to get started. We had a couple more people in here, but we're going to just go ahead, I think. I'm Ita Berner, and I'm coordinating this series it's our Informatics Summer Series that we've expanded a bit from previous years. Our speaker, I'm just going to introduce our speaker today and let him take over. Our speaker is Jake Chen, who is new to UAB. You've been here how long?
2.5 months.
2.5 months. At our newly developed Informatics Institute, which has been here for about 15 months or so. And Jake is Associate Director of the Informatics Institute and Chief of Bioinformatics for the Informatics Institute. And although his area of expertise is bioinformatics, if you remember, those of you who were here last time, we talked about biomedical informatics covering a full range of different types of data. So he's going to be talking about, let me get that big off there. He's going to be talking about biomedical data, which will include bioinformatics data, but also a broad range of data and the importance of structuring it and how to do it. Is that correct?
Yes.
Okay, Jake, why don't you take over?
All right, so feel free to stop me if you have questions. I do have slides, and I can go fast and slow depending on your interest. But people online, feel free to enter the text into your chat box, and if I didn't catch it, then people can alert me to it. Right? So, I mean, I'm not going to talk about the whole spectrum of bioinformatics, but only a very small part. Essentially, what is the biological data management? Why is it important?
I can always use this.
So I think many of you have already come to this idea of a high-throughput biology when people are talking about genome biology, and they're really interested in understanding sets of genes at a time and using high-throughput technologies such as genomics, proteomics. And then the difference here, there was a question there. You don't get the sound, is that what it is? Or maybe there's... Oh, okay. So it's just some self-introduction. So there are many different high-throughput omics data capturing devices, such as next-generation sequencing, microarray proteomics, kinomics, what name you. These devices capture lots of data all at once. So you cannot really do the traditional hypothesis-driven biology, which start with one gene, follow the genetics, biochemistry, and validation. So all of a sudden, you're dealing with lots and lots of data, and that's part of the reason that we need to talk about the data management. Just take one example. I'm familiar with the example that you have. In proteomics, and people are thinking about proteomics as having some mass spectrometer or some gels, and then you take the proteins and run through the gels, several dimensions, and then you carve out the spot. That's the old type. Right? If you look at the time frame, on the y-axis is actually the number, the amount of data that you can capture is just increasing really exponentially over the course of the time. And so the biological data are being generated in high volume and very fast speed. What are the characteristics of these biological data that differ from the online clickstream data you're familiar with when you're doing online shopping or e-commerce? I think these are some of the characteristics. One is that they're really, really broad. Right? In order to understand the complex human biology, you cannot afford just to study the gene. You have to study the epigenetics aspect. You have to see the variations as well as the copy numbers, and you have to look at the mutations as well as the gene expression. You have to study the transcription level, and then you have to study at the translational level proteins and post-translational modification. And you have to study the omics aspect, and then you also have to combine it with the clinical data, imaging data, and so on. So it's very, very diverse. And in this school of medicine, how many departments are there? How many centers are there? It's countless number, right? But if you think about every department, every center are producing their own type of data. And it's very, very complex to capture all of them and try to make sense of them. So we're a big world and bigger than the online community of shopping data. And then also every community would have their own special needs. And I would imagine people coming from cardiology would have completely different interests, and then not understanding the data from people coming from neuropsychiatry. So you cannot really build a software system for one community and expect it to work for another community. It's just like in the business world, you build an accounting software and you say, well, the finance department can also use it for projection. That's not going to work, right? And then all of these are actually experimental data driven because that's where the large data sets are coming from. And then you can say, well, I still have those survey data, and I still have those theoretically generated data. Those data are more or less we can manage. You can use your Excel spreadsheet, that's fine. But these are not really suitable for an old set of tools. And then you have many data types. Right? So DNA, RNA, structure, functional assays, drugs, and all of these data types need to be related to each other. So it's very complex, and they're also rapidly being accumulated. On the left side, you can see a figure that says the growth rate of the ENVO GenBank, and this GenBank is a repository of DNA information. Over the years, you can see that this information is growing at exponential scale. Right? I still remember that in 1984, well, I wasn't in the field. I started the field in 1995. Bioinformatics isn't called bioinformatics. My PhD advisor couldn't even pronounce the word. And I have to actually teach my PhD advisor because that at the time is called biophysics. We only have limited protein structure data, and the entire GenBank only have a few thousand proteins related to structures. Now it's completely different. And also, when you're looking at the knowledge that we're generating on online PubMed, and you're also seeing that the past 50 years has saw a much faster rate of accumulation of literature than in the previous time. So we're also generating knowledge faster. Right? So as a biologist, and many people are thinking, oh, I can manage everything through spreadsheet, and I'm a guru of spreadsheet, and even maybe Microsoft 2017 version allows you to have a limited number of rows. if you have a 60-bit processor. But is spreadsheet the solution to all your problems? And I think that if you believe in that, and I think you're falling into a trap, and the data is coming at a speed, it's not just a volume, okay? It's coming so fast, and you can barely keep up with the storage aspect of the data management, but it's very difficult to store it, process it, clean it, model it, analyze it, and then share only the useful sub-portion of it. So I think the big data indeed will become really big, messy data if you're not prepared. So the aims of biological data management is very straightforward, in my opinion. It's easy to understand. We need to be able to process and store these data in ever-increasing volumes. Right? So when we talk about big data, people like to say, oh, there are 4 V's. And nowadays, if you talk to even government officials, and they'll be able to say, well, what are those 4 V's? Volume, lots of data, velocity, speed, and variety, lots of different heterogeneous domains, and veracity is the truthfulness of the data is more important. But I think in biology, we have all of that, and especially the last two, veracity and the variety of these data. And then we need to process all of them and store them in a place that we can efficiently retrieve. But retrieval is not the main goal. If retrieval is the main goal, you can actually go to Google Life Science. Imagine you can build a database of everything and then you Google it. And then we don't have to do any biological research anymore. The problem is lots of the information that we're interested in are the association. What is the reason that this gene transcription factor regulating a target, and somehow this target is causing cancer, right? And so those information, you have to correlate the information and then model the data before you can actually query them. And none of the information retrieval systems are smart enough to tell you that. So you have to not only manage the data, but also manage the data for the purpose of modeling and interpretation. So in fact, I would say that extracting useful scientific knowledge from successful data management is extremely tricky. For people that can do it and people that can't do it, it's almost like the difference of magic, right? So for example, I have this cup, and if I can tell you that I can levitate, do you believe me, right? So look at this. How did I do it? Anyone? Well, I can let it levitate. Yeah, so actually I poke a hole and I put my finger in it. But informaticians are like magicians to some extent. We have lots of under the scene tool development behind it so that we make something that looks like magic so easy to you as a biologist. But actually there's lots of thoughts and requires some ingenuity to get it done. And then the last aspect is we need to share and disseminate the data and knowledge to the scientific community. NIH is very big on that. And I think in the world, the European countries and the US are really emphasizing that. They're essentially saying if you're funded by NIH, you're required to release the data to the community 9 months, roughly, after the data is deposited into a public database. And this is very important for the community to actually analyze this data and also contribute to the overall scientific realm. So we have biological databases. We don't have one huge homogeneous biological database. In fact, we have a database curated by different groups throughout the world. This is a growth of biological databases from 1999 to 2013. You can see that we have a steady but not exponential growth. This is because creating databases are very labor intensive. And these are just 3,000 of, I think 2,000 of them are just a published database portion by nucleic acid research. And there are lots of unpublished databases, and I would say that there's probably today 5,000 to 6,000 biomedical databases that are open in nature in the world. And if you think about those databases, they capture many aspects of information. And here I have to focus on bioinformatics because that tends to be more open. If you try to look at their categories, lots of them are the classical so-called sequence or structural databases. For example, I'm going to talk about GenBank, and GenBank is a collection of all of the sequences that have ever been known from different sources. And then you have protein structure database like PDB that has all of the NMR, X-ray, or even some theoretical predicted protein structures. In fact, it's so important that the guy who was behind the PDB, Phil Bourne, my colleague that came from UCSD, is now the NIH Associate Director of Data Sciences. He reports directly to Francis Collin. That shows how important that this biological data management is to the overall NIH overarching goals. And then you also have these so-called genome databases that are more comprehensive, like Flybase, SGD. These are all more or less focused on a specific organism and very comprehensive in the content. And there are databases that are functional genomics in nature. People load the high-throughput data from microarrays or RNA-seq. They annotate it by gene, by transcript, and by tissue, by organism, and then they really make it easy for you to search. And there are systems biology databases that is my favorite, that is not meaning try to look at the different structures or components, but look at the relationship between structures. For example, relationship between genes and genes. What are the co-expression patterns? Or protein and protein interactions, or protein and drug interactions, and the pathway and pathway similarity. And there are so many more different kinds of databases. And usually, if you want to go to these journals, Bioinformatics database or nucleic acid research, you'll see that these journals would try to compile peer-reviewed and well-publicized resources. Some even have just occasional updates.
All these databases, is there any security or vulnerability to us for them being, would anyone ever want to hack and mess these databases up?
Right, the question is, are we concerned about the security for the biological databases? I think the security is mostly a concern when it relates to human sensitive data. For example, there's a database called the DBGAP, and that contains a genome to phenotype, genotype to phenotype association data. It's coming from real clinical sample with patient attributes. And for information that's sensitive, they do classify information as tier 1, 2, and so on. And they only release to the public the summarized information. Likewise, for TCGA for cancer, they do it that way. You have to actually provide your own information, gain consent, and agree to destroy the data after you're done with analyzing in order to have access. But in general, if there's information out there and people are not too much concerned by the sensitivity. In fact, if you're a terrorist and if you have all the knowledge, and you can actually build your terrorist weapons. But I think this is the same with all the technologies. We can't stop the evil use of the technology, we can only set up standards and promote the good aspect, hoping that the good aspect will outperform the evil aspect. That's sort of the spirit for biology and biomedical research community. If you go to business, if you go to chemistry, and it's actually quite different. In chemistry, I think that lots of people don't like to share, other than the PubMed, which is also driven by the biological community. And prior to that, for many years, and they even charge for accessing those information. Okay, so we have lots of databases, but I think the problem is just getting messier. What I'm showing here is how these databases are linked together. Imagine you're a researcher, you're trying to get to one database, ask questions. Do you want to go to 1,000 databases to ask a simple question? What is a favorite, I have a favorite gene signature. What is everything that I can know about this relevant to any diseases? You may have to go to so many databases. So maintaining these databases is very difficult. You have to rely on the community to maintain it. Some groups are well-funded, some groups are not. Like the UniPro database, they receive an annual funding of several $1 million, and there are actually about any given time 100 curators throughout Europe and the United States to curate those content. But then my database, published in my own research group, and I only have maybe $50,000, and I have to be really quick, and I have to be solving a very particular problem. But after the students are gone, sometimes the database may not be just maintained well. So you have all kinds of problems, inconsistent data formats, because people don't do it to the fullest, and they're not professionally trained. And they have sometimes inconsistent semantics. For example, what you call gene may be different from one database to another. And it's also because our concept of genes evolved over time. If you go back 20 years, people say gene is protein coding genetic structure. But if you go back 80 years, and you don't even have to have a nucleotide sequence tied to it. It's just a concept, it's a genetic component. But if you fast forward to today, people will say gene could be RNA coding, and that doesn't have to be translated into proteins, right? And it can be all kinds of things. And you may also have redundancy information. And when you have redundancy, it will be great. Very, very lucky that you have redundancy and consistent information. Usually redundancy means they disagree. What do you do when you see different databases have different content? And there are gaps in the knowledge. Anytime you want to go and ask the question, do I have the predictor for the aggressiveness of brain tumor in any database today that I can act on and design a drug for it? I can tell you there's none. Right? So those gaps are very, very important for scientific discovery. And it's difficult to navigate, and so user interface issue as well. So I think I have some more details, and I'm going to actually skip some of the content. But you get the idea. It's very heterogeneous, and is very inconsistent in the format. Those are just often discussing geeky terms and the XML, relational format, NoSQL format. And those technology changes over time, depending on at what stage of computer science or IT you're just implementing your system. And in the old days, and people are proud of managing data in COBOL, right? And I learned my first computer science in Fortran. And but today, I think if you go to any high schoolers, and they would tell you that probably the Web 2.0 technologies and programming to the semantic web is the latest thing. And it's very different if you're dealing with the legacy data and try to make it consistent. Semantic, we already mentioned about the semantics of the genes, and they're very different. And then the worst part is that the content is evolving dynamically. So think about if you're in an e-commerce setting. At Amazon, if you say you're trying to buy a dehumidifier, and all the descriptions are the same, right? So you're not going to have the dehumidifier one day it says, oh, it's going to actually extract moisture at 50 pints per day, and then the next day change to 3 or 4. But in biology, we're discovering this information. We can actually update the content. When you update the content, imagine if there are people publishing paper, do they want the content to be changed? They don't. So there's a change management, but there's a natural nature for science to evolve over time. So it's very, very complex. That's also says that lots of people are quite interested and fascinated in the managing the biological data. So I think it's proper for me to introduce some very basic concept for you. At least that this is useful for you when you're interviewing and say, well, I learned some informatics, so at least that these terminology is important. They're different. So you don't want to talk about the database and confuse it with database models, or database management system. A database is passive. It's a collection of data usually that follows a specific set of structure. If you're talking about relational database, usually it's described by a mathematical formulation of a basic data structure called relation. It's not a relationship, right? So it's a basic data structure called relation, and then you implement that relation as a basic data type, and then for those kind of databases, implemented as called relational database. But there are so many different kinds of databases. XML database, there's no Excel database, unfortunately. A database model usually describes at a higher level what is the inherent structures behind all of those data, millions of data points. Imagine you have a sequence database, you have all the transcription factors listed in one table. But the structure of those transcription factors could be very simple. The transcription factor would have a protein ID, would have a binding the motif, and would have maybe tissue where it's highly expressed. These 3 attributes will be the essential data structure no matter how many data points you have in that database. But imagine that this is not the only information. You may also have the motif, you may have the species, and then all of those structure is what the data model is trying to describe. And then there's a concept of database management system. It's the software that manages your database. It's the specialized software you don't want to write. And people have won the equivalent of the Nobel Prize in computer science called the Turing Award for coming up with efficient ways of just doing database management. And so Michael Stonebreaker, for example, is one of the person that I really admire. And he came up with many databases, Postgres, Prepress, and so on, and even the current days new SQL, NoSQL databases. Made lots of contributions. Usually those people are working on the software, but as an interdisciplinary biomedical scientist, you don't want to work on the software. It's very, very tricky, and you just want to use it, right? And some examples of these are MySQL, Oracle, MongoDB, ArrayDB, and those things. A database system usually, when people use the term loosely, it means a database plus database management system. Or database applications is actually everything, including the user interface. So when someone in your lab say, well, let's create a database, the first thing you need to understand is, Do you want to just create a structure so that you can store the data regardless of how difficult it is to retrieve? Or do you want to create a whole set of applications along with the licensed software, along with the staff, along with maintenance, along with the ability to interoperate with other applications? And it's completely different games from one to another. Right? So when people are, right, for example, go to the BERT and try to say, well, do a database design, and sometimes you may get the idea, oh, how come this other party told me it's $1,000 to create a database? How come you charge me for $10,000? Right? They're very different. Wouldn't you agree? Yeah. So we need databases. I think these are some more detailed information. You don't need a database just to keep a persistent storage of data. That's a very important aspect. What do we mean by persistent? If you unplug it, will the data still be there? Well, it's possible it's lost. Or if you have a hard drive failure with the database, will the data still be there? I mean, if you think about it, if the building is on fire, will the data still be there? Right? In 9/11, there are actually companies that have the off-site backup that actually benefited, and then they come back very quickly. But there are companies that have their data vault right in the basement of the building, and then it's still it's not useful. You need to think about to what extent can you tolerate the failure and design it in a way that's appropriate. And then you also need to do some bookkeeping. Bookkeeping means that the database is not stored once and for all, never touch it. And every day you say, well, I'm going to pull out a record and enter some information. This is what is called a transactional information. Right? So every day you type in something, and then the database needs to make sure that the information you type in is consistent. It's not going to change. For example, if you are trying to take $10 from your bank account, and if your spouse is trying to deposit $50, if the bank doesn't have a good way of resolving this, the conflict, when you two say, well, let's time it, let's do it right at this exact second, do you think the bank will either lose or gain money because of your timing? No, they're not going to lose money. They're going to log the record, make sure that only one of the transactions goes through, and so that you're essentially not going to outwit the system. But so that's part of the bookkeeping. Abstraction and installation. In programming, we often need to think in abstract terms. You don't want to see the so-called the trees when you're thinking about a general pattern. So at the when you're flying on top of the large forest and looking for areas of insect infections, you don't necessarily just go count the number of insects of every tree, right? You probably just look at the very high level the structure of those diseased trees, and then you get a pretty good idea. Likewise, in data management, you don't necessarily look at every piece of data, but you want to look at the statistics. That's why we have the the BERT there. Reconfigurable views. And the database is very difficult, it's very challenging to manage, and you want to actually invest once, and then you want to actually use this to build different applications. So a very close example is at the clinical record. So we only go to the hospital once, but the clinical record that we have may be built into many different applications. May be used by the Department of Public Health for epidemiology studies, or it may be used by a particular disease study, or it may be used for genetics study. So every study you can imagine is taking snapshot of parts of the data and then combine them together. So this is what I call the Chinese herbal medicines way of doing business. So I don't know how many of you have seen a real Chinese herbal medicine. There are probably about 100 ingredients, and they're filed in little cabinets and little drawers. And then if you say, well, I have this sickness, and then they will take drawer number 110 and drawer 205, and they will concoct it in different proportions and say, here you are, and then you're actually cured, not knowing exactly why it works. But if you have any disease, and then they can do just a gazillion number of configurations. So it's very fascinating. Likewise, in database, if you actually decompose the information to a certain level of granularity, you can reconfigure it and allow the application to answer any question you would have. Right, for example, I would have the skills by looking at any website today, even at Amazon or eBay, very, very sophisticated system, and I can actually reverse engineer it. Right? Because I can actually look at the views and think about the data pieces and think about how to implement it. But I mean, that's a skill that eventually I hope you have. Data sharing, and then essentially have a web interface to it. To turn the data into a database, you really need to think about, well, do I care about keeping the data? Many people say, well, I don't. The raw data is too big. I'm only having $100 for the analysis. Why do I need to be bothered with it? But are you 100% certain that the cheap one-pass solution will generate the outcome? If you're not, you better actually be very careful, because then your graduate student, or some of you are a graduate student, may choose the wrong parameter when running the program. And by the time you find it out, and you may actually go back to the source and redo it. So you want to keep the raw data, but you don't necessarily keep everything. When you're dealing with a collaboration with Hudson and Alpha, and they have all the raw data, even the imaging files, prior to generating the FASTQ file. Those are analog data, and they even throw away those imaging and sequencing data. They can't afford to store it. And so there are decisions to be made at what level you want to make them into a database. And then you want to actually manage it by adding additional attributes, tracking additional information that the experimental data itself doesn't give you, like a curator. Who is a curator of your information? So next time you can go find him at lunch and say, hey, there's something wrong with the data. And then you should support multiple user high-performance access, and those are pretty obvious. And the support flexible reports based on the views I just mentioned. Distribute your data, then obviously you need to make a web portal. I think a DBMS, and we more or less mentioned that, the DBMS is a very sophisticated piece of software that allows you to specify instead of program to manage your data. You can, a very typical scenario to say, select what attribute from what table where the restriction condition is blah, blah, blah. You can just write this simple SQL sentences to do the equivalent of something like 100 lines of Java code. And the DBMS is really meant to make things easier, and also have lots of permissions and security features that make sure that only the intended access is allowed. And let's just, for example, let's look at one example. Here, you're trying to write a syntax select the book ISBN title, price, and price times 0.6% is the sales tax. But what is the sales tax here? Probably more than 6%. And from book table where the price is greater than $100 and ordered by title. This would be one example. Writing something like this can be trained and learned, but we're not going to teach you here. If you're interested, you can read about the SQL later on. And you can also create a table. You can insert content in batch. You can have this concept of transactions to make sure that whatever you have created is saved permanently, not lost due to the power. It's called a commit. And you can even update as what we call the bookkeeping features, bringing out the record and update some content. So this DBMS software, such as MySQL, Postgres, Oracle, and they all have different characteristics. MySQL is completely free. Oracle is the most expensive. What is the difference? I think the difference is in the performance. If you only have 1 million data points, and probably you can do away with Postgres. If you have 1 billion data points, you probably want to have Oracle. If you have 100 billion, you probably want to use NoSQL database that will push the limit. And so we're still pushing the frontiers of the DBMS software today. For database models, and I would like to also mention that there are conceptual models that people use to capture the structure. For example, this is a hierarchical model. What you see here are just graphical representations of these conceptual models: organization, department, program, and the language. So this is a top-down level to classify the structure of the organization. And here we have the school, department, center, but I heard that the center can be affiliated with multiple departments. So this, the UAB structure will not be described well by a pure hierarchical model because you will have multiple parents. But here, this kind of model, it only allows for one single parent. For a structure like this, usually when you see people talk about the document, which is architecture DOM model, XML, they're really convenient for XML to be expressed in these hierarchical models. Because it's a document, you can embed an element within element. So those kind of relationships are hierarchical in nature. And then the network model is a lot more complex. So you look at this, you see, well, this is an RDF semantic web representation. Exactly what it is trying to show the detailed relationship between data pieces. Therefore, you have somebody's name is Jack, and the address is in the doghouse, and in the particular street address of 435 Hoffman. And then and then some other things have a name Lumpy, and then there's friends between Jack and Allison as a home that's also that's a next door neighbor to Bob. So this network model allows any kind of a complex relationship between objects. And this is as complex as it can be, but it's very difficult to navigate. In fact, in today, if you're using NoSQL, or if you're using the next generation of a web called the semantic web, not just based on hyperlink, but based on understanding what the links will imply semantically to navigate those web. It's called a semantic web. And the data is represented this way. Right? So it can express fully the information out there, but the problem of this model is that it makes querying and mining information tricky because you're only seeing instances, but bags and sets that makes retrieval efficient. So there's some kind of the efficiency model usually implied by using network model. And then there's the object model that's usually used in the software world. I won't say it, but relational model is something that has been around for 30-some years, 40 years, no, 40-some years. 1971 is the paper that described relational database model. And it's very flexible. In fact, if you see most biomedical databases or ontologies, the underlying model are usually expressed in relational data model. For example, you have products and the order. And the product and order, the relationship is many to many. A product can go into multiple order, and order can consist of multiple products. And this is a model that describes the many-to-many relationship between them. Now I want to ask you, between gene and the mutation, what is the relationship between gene and mutation? Is it many-to-many, many-to-one, one-to-many, or many or one-to-one? Don't know? All right, so one gene, you can have just one mutation, you can have many. So,
just be many.
yeah, so from gene to mutation is one to many. Now let's say for mutation, can a mutation be shared by multiple genes? Probably not, because the mutation, usually we say that the mutation is, there's a context. And no matter where you find that mutation out, maybe it's due to your inability to resolve the sequencing reads onto the genome, but the mutation is a fixed concept. So in that case, it's not many to many, it's many to one. So, but sometime when I did the non-biological ones, and I asked them the question, what is the relationship between the marriage relationship between men and women? And you can trigger many kinds of many to many or one to many. I didn't say that in Saudi Arabia. I gave a lecture there. And I think that what we suspect is real. Anyway. So database applications, you can have data management and all of those things that you use. For example, this is one database application called TCGA, and you can have a web portal and you see all of these elements. It's actually not just the static web page. The fact that they have all of these elements and choices really suggests that they have an underlying database that serves the content dynamically, right? So if you were to hand code this information, then I think that you lose the point. You will be spending billions of years just to get the complexity of the application. So let's just take very quickly and look at one example of GenBank and RefSeq. So GenBank is a database curated by the National Center for Biological Information, and the RefSeq is a subset that's relevant to GenBank. What GenBank aims to do is to try to become a clearinghouse of sequences and their annotations. It is the world's collaboration between Japan and European countries called EMBO, EDBJ, and the US and CBI. And it's got as many species as you can imagine, but this was old times, and 6 years ago they already have 100 million sequences, but I think today they have more than quadruple that information. Lots of completed genome, not just human, microbial genomes, fully assembled, and eukaryotic assemblies. But for lots of the genome, and many uncharacterized microbial genome, it would simply not have the complete genome, but just fragments of sequences or assembled contigs as they arrive. Right, and then for the organization, you can have different naming standards. You can go to the database and learn about it. But one thing that you know is that GenBank being so old, they don't have a good representation. They have what is called the ASN.1. It's a precursor to relational definition, but it's not relational. It just is like an Excel spreadsheet. They have a standard vocabulary terminology, but it's very flat, or at most a hierarchical model to describe how the data element should be embedded. So this is a printout of the ASN.1 format. You see the locus, the gene name, definition, accession number, and so on, the journal references. It's tricky to actually write to this format. So eventually they actually converted to relational format. But it's not just a storage unit. So people can submit to GenBank. If you're a sequencing center like Huffman Alpha or WashU or Baylor, you actually have automatic script. Once sequence or sequence, and then you can deposit into GenBank, waiting for annotation. So there are tools called Bankit that allows you to batch submit information. What if you machine went berserk and submit lots of round sequences? Well, there are mechanisms to retract sequences. A RefSeq, on the other hand, is a curated, redundancy removed subset of information. It tries to give a better knowledge view of all the information. So it's going to be much more focused, and curation is the key. But curation doesn't necessarily mean always a manual curation. It can mean that corroborating from multiple sources and use computer program to curate that. And there's a whole pipeline that people use to curate information, annotate information, and extract information for different elements of the annotation. Say if you have a very good post-translational protein, which is a functional predictor, you can actually suggest to RefSeq to use your predictor if your work is highly established. So it's really a community effort. And I listed some differences between the tools, and you have the slides. So if you want to access it, they actually provide the web user interface. You go to NCBI, you say Entrez, and you can type in some sequences, you'll get the result back. But this information, this type of data management, as I mentioned at the very beginning, is primarily for fact retrieval. So in order to compare sequences, you need to actually come up with better tools like BLAST or a multiple sequence alignment tool to analyze this information. So as you move away from a regular user in biomedical informatics, which these tools will become very essential, to an expert user where you know all the nuances of changing the parameter, to a tool developer, a person like myself, we actually don't use these tools very often. So in fact, you can learn to become better than I am in using the latest version. What we usually do is to look at maybe several iterations along the way and say that what GenBank is still not able to do, probably GenBank is still not able to figure out the relationship between protein sequences and what are their co-expression patterns. And then we do research and make sure that we build the tools to eventually help people answer those questions. So those are really fascinating to me, but I think it may not be always fascinating to yourself. But anyway, so I think we're exactly at 4 o'clock, and maybe we can have the luxury of a few questions. You're welcome to send me email or arrange a time to stop by and say, well, hey, I've got questions, I'm completely confused, and it's okay to try to find them. My email is jchen@UAB. Any questions? I know usually this no question means that everyone is so hungry and exhausted and not understanding. So can I have at least one question?
So can you change the network model into the hierarchical model if you have like you're looking in one area and you want to make that your center? Can you change
Yes, yes. To change from network to hierarchical model is possible if you have actually a particular linear order between things. And then by doing so, you will introduce some redundancy because their relationship as naturally is cross-linked. What you do is to simply stick the same information twice or multiple times in the hierarchical model. You can do that. You can easily convert from hierarchical model to network models. All right, with that, and I think that
let me send that on the mic and go up to the print. Thank you all, those of you online as well as those of you here. Next week, Dr. Jim Semino, who is director of the Informatics Institute, will be speaking. I won't be here, so he'll introduce himself, and then I'll be back afterwards. But after his talk, there's going to be an online session. And basically, that's one where I will mail all the people who are at the talk, at Jim's talk, and anyone else who can't make his talk but who wants the online session should just email me. I will basically give you links to view some presentations, and we'll have a little exercise for you to do online. And then the following, and there's a break over the July 4th holiday, so I'll try to get out to you sometime. Well, this talk will be available. I'll give you the link for Dr. Chen's talk and give you the readings probably on Thursday. I'll be emailing that to you. And then I'll email you after Jim Semino's talk, and then sometime shortly after that is when I'll send you the information on the online session.